How can we grow muscle to reverse degeneration and promote health? Steroids and drugs like Ozempic have many downsides. Longevity drugs and cures and all these things that you read about or hear about in the news or on YouTube seem really far away. What if there was something exciting going on right now that you could learn about in this episode? We'll meet Matt Scholz, serial founder of Oisin Biotechnology, targeting senescent cells for age-related pathologies, cancer cells for solid tumors, and muscle growth, as well as the founder of Immunosoft Therapies, which has the mission to create novel therapies for rare diseases.
So we focused on delivering nucleic acid payloads that basically put a suicide gene under a version of the P16 promoter. And so it is actually quite similar in principle to that and very different than a drug-based approach. The beauty, though, of our approach is that if you want to target another pathway with it, like say P53, you don't need to build a whole new drug. It's just another bit of code. The phenotype is 4 times the muscle mass. Massive.
He did all that, and he's not even a doctor. Oisin is named after the Gaelic warrior bard of the 7th century, which sought the fountain of youth. Matt is turning that myth into reality and giving us a behind-the-scenes look into his company, his practices, the way he does business, as well as the way he does science, including revealing how he landed Tim Draper, one of the biggest investors in Silicon Valley in the United States as a whole.
Before he got on stage, the newly elected as the prime minister of Austria. So it's like a high-profile event, and then he gets on and does this whole thing. And I decided the best way to corner him was to...
We'll need to watch the full episode to find out.
Learning with Lowell. Learning with Lowell is not recorded in front of a live studio audience, but we do take any questions. Stay tuned and stay curious.
Oisin is developing a high precise, patent-pending DNA-targeting intervention to clear these cells. As a recent study has shown, clearing senescent cells both reduces negative effects of aging pathologies and also extends median lifespan and survival. So that's a quote straight from your website, which aligns with a listener question. A shiny bauble says, there are dozens or more senescent cell companies. Most have struggled to find a mechanism that is sufficiently safe, selective for senescent cells, to use therapeutically, while also providing good drug-like properties. What makes Oisin different?
The fundamental approach we've taken to this is quite different than I'd say most, if not all, of the companies out there. And that we set out to basically kill cells based on what they were thinking, not what they looked like or how they were acting. And but more technically, we're killing cells based on their transcriptional activity. And this was really largely inspired by a study that I think, to some extent, launched the field. This Buck Mayo Clinic study where they made the transgenic mice that allowed them to kill P16 positive cells with an otherwise innocuous drug. And the results for that were quite profound, but I suppose in the tradition of a lot of these kind of academic studies, it wasn't translationally useful. You can't go back in time and modify your own embryo, become a transgenic organism. And so for the most part, people have gone about trying to kill senescent cells with repurposed chemotherapies. And yeah, it's a fair point. Making those things selective and safe enough is a real challenge. When we started Oisin, like our goal was to actually make a clinically viable version of that transgenic study. And so we focused on delivering nucleic acid payloads that basically put a suicide gene under a version of the P16 promoter. And so it is actually quite similar in principle to that and very different than a drug-based approach. And so and I think it's a fair point to say that, like, P16 positive cells are not like, a perfect overlap on the Venn diagram of senescent cells. And I think it's also fair to say that the field at large has not yet fully characterized every kind of population of senescent cell. Like, there's still ones I think we don't know exist. And whether getting rid of them is makes things better or worse is actually still an open question. But P16 was a pretty good one, and in those studies, even wiping them out, weekly from the time they were weaned, didn't seem to have any deleterious effects. And so we thought it was a relatively safe and effective way to go about it. The beauty, though, of our approach is that if you want to target another pathway with it, like say P53,
at any point in this conversation, if you find value in it, please subscribe. It is hugely beneficial, and it tells Google and everyone out there that this is content worth watching. Thank you for everyone thus far who has commented, liked, subscribed, and told their friends.
You don't need to build a whole new drug. It's just another bit of code. The North Star really for Oisin in general going through this is that the essence of life is information. And if you can treat diseases with information, you can do things that really would be impossible, if not wildly impractical, at least, with conventional medicinal chemistry. And so to go after P16 and P53 is trivial. Like there's no need to re-engineer everything. It's just another sequence. And as you start looking at new pathways as the field moves along, I think we can adapt to that in a way that it's very hard to do in any other approach. And so the other thing, we haven't seen toxicity from this that looks problematic. But if you discovered say like P21 is a good example of one that looked like it might have real toxicity if you use that as a primary targeting pathway. You could put what amounts to Boolean logic in your construct because it's now code. It's just DNA, and so you could put a microRNA target or interfering RNA or a suppressor under, and you could actually restrict its expression in tissues in a way that you really couldn't do practically with other approaches.
So I have not been in a lab before, so I don't know the difficulty of actually engineering these things. How hard is it? Like the process, what's that process like? If this is like 2IP, it's fine to just do like a high level. But the Cas9 suicide gene, it reminds me of like Cas9 when they with mosquitoes that they engineered the gene in there, which caused like a gene drive in it, so that like the next generation will all become male, so they all die out, so they can't populate out. That's what I assume is like a structure. But what is it like for what you're doing?
Well, so I guess I should make a distinction probably more for the audience than anything else. But Cas9, as in what's used with like CRISPR type stuff, and Cas9 are actually very different things. Like one is making double-strand breaks. The other is a suicide gene, but the acronyms are similar enough that I frequently get this question where someone's mixing them up. But I guess that a real fundamental difference with the way our technology works is that none of these things integrate into the genome of the organism. So they're DNA, but they're episomal. So they exist as basically a plasmid in the nucleus, but not in the genome itself. And so the goal is not to say eradicate the senescence pathway, which has a lot of utility. The goal is just to make a point in time intervention where those cells will die and other cells will be unharmed, and then you can make a new senescent cell the next day. And so but what's really fascinating about how the field's evolved is we're at a point now where you can type a gene sequence into your iPhone and it'll show up in the mail in a FedEx envelope. Like you can write a program in the language of life and get it delivered to your doorstep. And so the real engineering challenge in the lab and in life is delivery. Your body has a vested interest in not letting foreign DNA execute. And so it puts up a fight. And so you want to basically be able to evade those defenses without causing harm. And traditionally, we use viral vectors for this, right? Adenoviruses, gamma retroviral vectors and lentiviral vectors and all these things, AAV. And they have a real issue in terms of your body knows that a virus is and doesn't like it. And most of them can only be used once. And a lot of them, they carry all sorts of interesting constraints. But their benefit is, of course, they're a purpose-built machine optimized for nucleic acid delivery. But let's see, your body is aware of these things and has been at war with them. Ever since life existed, it's been locked in a struggle with the virus. And so ours is a non-viral vector, and it doesn't have that limitation. And so you can administer it at really high doses, repeatedly, safely, and you can deliver these payloads across the body. And I think it's a kind of a unique problem, but it's really led to me developing a professional obsession of sorts with nucleic acid delivery. I think that is really the choke point for these kinds of therapies.
How does a nucleic acid delivery compare to like mRNA vaccines that people are more familiar with when it comes to like COVID and stuff?
Well, there's some overlap in them, really. You can use if you take the COVID vaccine, some of them were actually full-on DNA viral vectors, like adenovirus one, things like the J&J one or the AstraZeneca one. Those were both a straight-out DNA virus. Most, like the Pfizer Moderna ones that were most common in the US, were lipid nanoparticle based mRNA. And the technology we use, the proteolipid vehicle, is capable of delivering mRNA. So you could actually do this with the same technology. If you put DNA and go the other way, but DNA into a lipid nanoparticle becomes far more toxic actually. So they're not used for that. But our particular one can go either way. And the reason we're using DNA is we want to take advantage of the control logic that it affords. So if you put mRNA into these things, it'll go to any cell and it will execute. And if you do that with a suicide gene, well, bad things are probably going to happen. It's going to think Indiana Jones face melting type style, where like any cell that's contacted just dies. So we want the control logic for this particular application, but we can use mRNA for other things.
Makes sense. Outside of the science, one thing that I'm always really excited to learn about is like the business side or the other leadership areas that I think a lot of people, when they're reading about the clinical trials or they're reading about the new IP that's coming out, they don't realize that there's a lot of other things that need to work or for the science to even make sense. I was working at I was once working with a deep tech company. I'm trying to like be nondescript because I'll be disparaging in a second, but they were once on the outside I was like, oh, okay, these people know what they're doing. And on the inside I was like, oh wow, like they tape everything down. And so but there was like kind of like leadership issues and they eventually died, and you kind of see it coming from the inside. And so I'm always curious to learn about the things that people don't realize. It's not always just the science, like things need to be right for the science, also need the leadership and the finance and stuff. So what do you do anything interesting when it comes to setting up the business or structuring like your burn downs or anything like that? Where I think a good example of this is the recent interview with Kelsey Moody, and their business model is really interesting because they're profitable, so they can keep like innovating and stuff like that. So when it comes to the non-science side of things, what's like some of your differentiations, considering the fact that you're like 15, 20 years, you have a long experience to pull from to like do things differently?
Yeah, I think part of the key here is figuring out where you fit in the ecosystem and what you're good at, what you like, and that should dictate how you build your teams and structure your organization. And for me, I'd say if you were to distill it down, I'm really passionate about taking things out of the lab and into the real world. I like finding really cool pieces of technology and figuring out how to build them into something that's therapeutic and useful. So it's like this kind of early translational edge. And so we're not likely to, we're not going to be a commercial pharmaceutical company anytime soon, although I would like to actually at some point do that. So our teams are typically very small. They're focused on finding people who know their spaces really well. Like a good example probably of this was the first one I started, Immusoft. And before that, I had no background in bio, and I licensed some technology out of Caltech and later out of University of Minnesota, and then had found some weird stuff in France. As I started to build a lab for the first time, ever, and bring people in, I said, look, we're going to be the undisputed world experts in this technology. In this case, producing engineered B cells, autologous long-lived plasma cells to secrete therapeutics. And I said, we're going to know this better than anyone, and like we'll know better than like all the experts out there, like from the tops of their fields to the journals to the regulators. Like this is going to be our expertise, and we're not going to do anything else. Like if it's not a part of that, like we're going to outsource it. And so we basically ran it that hard for like 7 years, and eventually, that went into clinic fairly recently, just in the last few months. The first human ever received an engineered B cell therapy. It's super exciting, but it built like a lead that I think is largely insurmountable. Like that level of focus, you didn't need to have 100 people and like $100 million. Like you needed to have people who knew that stuff better than anyone and could build it, and figure out how to scale this idea from like the first fluorescent cell in a dish. Actually, some of my early proposals and reports, like one GFP positive cell. I'm like, oh, look at that cell. It's amazing. It's a primary B cell that we transfected. And that to something that you can make at human scale for a patient in GMP conditions. And so I think that for my purposes, it's really trying to build like small kind of tactical, much more like I guess a SEAL team than a huge organization. It's trying to figure out who do I need to accomplish this particular mission. And I said, as you go out farther, I think that going after aging is a monumental challenge, and I think you'll need the resources of a big pharma to do it at some point. That's why I think I'd like to do that at some point, is to get big enough that you can actually run drugs all the way through to commercial. But that is such a massive challenge, and there's not like a lot of easy ways to get there. I mean, you need to first, like build things and license them out, have these successes early on and kind of build this platform and pipeline to do it. So that's where we focused. And I think that as a general rule, if you aren't looking at your environment and how you fit into it, it's really hard to put together a team that's going to be successful. And I'm sure, like I'm familiar with Kelsey's background. It's really fascinating. Kind of like, I suppose, in some respects, like mine, although he is more in the space than I was, but unconventional start. And you see more of this. I mean, the stats, something like 3/4 of all blockbuster drugs on the market come from companies that are small biotech, small pharma. So if you look at big pharma companies, they're a lot more like a record label than a band. They don't make drugs as much as they sell them, and they're willing to pay a premium to get them from little companies. But they've largely decided they're better at selling and distributing things than they are at making them. And I think that it's an important thing to understand that if you're going out to start something, like, how are you going to fit into this?
Yeah. So the focus and obsessing down on a specific song to make, and then building it up to an album that will then be distributed up to the record label. So the big things I heard were focusing and obsession. one I think opportunity to like follow on with this is you transitioned into the field. Like, you weren't in biotech originally. What did you do? Or tell me about how you got people to believe in you, I guess.
It was painful and iterative. I won't lie. It was a little bumpy. And so I guess the initial angle in was through cybersecurity, and I got interested. That's how I was in computer science background, and the thing that really first captivated my imagination was the idea of immune memory. And like, you get take like an 80-year-old who's been vaccinated against smallpox or got it when they were young. Like, they still have antibodies floating around to that disease, and they're made by the same cells that were there all those years ago. And so as the kind of computer scientist, I got wondered, like, well, where's the file? Where's the hard drive? Like, in what language is immune memory written? And it kind of led me down this rabbit hole in immunology, the adaptive or humoral immune system in particular. But and when I started thinking about diseases you couldn't vaccinate against effectively, like HIV, there's an interesting phenomenon, right? I mean, this is a disease that's killed tens of millions of people, and everyone who's got it made countless trillions of antibodies in an effort to fight it, and eventually succumbed. But a handful of people made antibodies that kept them safe. They either didn't get sick or they got sick very slowly. And they call these guys elite suppressors or elite controllers. And we've pulled the sequence for these antibodies out of the arms of survivors in Africa primarily, and began to like optimize them in a lab. But if you think about this conceptually, we waged a brute force attack with flesh and blood against a computer program, against a virus. And we fought this battle with the souls of humanity and against a computer program. And so I got interested in trying to use cryptographic hardware, like password breakers, like really high-powered ones, to effectively evolve the immune system in silico, to wage that war with silicon instead of flesh and blood. And so I'd started playing around with things like protein docking and all this kind of stuff early on, and long before AlphaFold and all these cool things. But as I got into it, some snarky scientists had pointed out that cells don't have USB ports, and you can't just program them. Like, you couldn't, like, we already knew the sequence for the broadly neutralizing antibodies. You couldn't upload them to people, and that kind of blew me away. And I guess it's a testament to my ignorance of the field. But I'd assume that, like, people already knew how to do that. Like, I mean, people have been obsessed with manipulating DNA ever since they knew what double helix looked like. And like, how could we be that bad at it? And it's like 15 years ago. There's no approved gene or cell therapies back then. And so I started that first company to build an app store for the human body, a way of brokering information into life, so you could specifically program the immune system. And that became the point of focus. It's like, I want to, I need to learn enough about immunology to figure out what cells you need to program and how you like identify them, capture them, manipulate them, grow them, all these things. And that became the thesis that guided all the technical development. It was probably a couple of years, I think, from the time I had this idea to the time I got funded. And it was bumpy. Like, I started out by just going to every bio event I could find and telling anyone who'd listen I wanted to build my app store. And the general objection was just, you're crazy. That's not going to work. And you have no business being in this field anyway. You don't know anything about it. And every time this would happen, I basically corner someone and say, okay, well, what's the biggest problem then? Like, what's going to break first? Why is this impossible? And then I'd start reading about that topic, and I'd find someone who wrote about it a lot who I liked, and I would call them up and say, hey, I'm trying to build my app store, and I think there's a problem. Could you help me? And I built this really cool advisory board that way by basically just every time someone had a problem I didn't know the answer to, I would go find an expert in it and read up all this stuff. And I do weird things. I read, like the actual dissertations people have written and things like that. Something that I don't think most people actually read their own dissertation all the way through. It's one of those things that's largely forgotten once it's done. And people will always be shocked by this. And for me, it's that I didn't know anything about the field. Like I'd sit around reading like methods in molecular biology, just protocol for protocol, trying to understand like what were the tools that were used, how did you actually do these things, and how did it work? And eventually people ran out of reasons why it wouldn't work, and we got funded.
So it sounds like one thing that you did there was you leaned into the difference. I think sometimes people feel like they need to hide the fact that they're not, that they don't belong or whatever. But it sounds like you not only ran with it, but you really made, even your language in describing it involves that cybersecurity background that you had. And so like you didn't hide it, like you ran with it, you pushed into it, and then you used that that different way of thinking as a differentiator. It sounds like when people were assessing whether or not to give you money, they weren't like, oh, is this guy on the level? It's like, oh, he's very transparent in what he's doing, which is pretty cool. And the other thing I was going to ask you, like, how did you learn? How did you learn enough to be sufficient? And I've never heard of this process before, where you crowdsourced the focus.
Crowdsourced rejection.
Yeah. It's like, you going up to someone and say, hey, this is my idea. It's like, oh, this is all the reasons it can't work. It's like, oh, great, I'll be back in a month. That's a really very targeted way to talk to the smartest people, tell them your idea, and then just watch how they respond to it. That's really smart. I'm gonna definitely steal that. If any idea I have for moving on, like that's very a repeatable process for anyone listening as well.
It's particularly effective in the bio space because scientists, they can kind of be dicks. They don't feel bad about making you look stupid. Some of them, like I'm convinced, get off on this. They they love it. There's a certain demographic I can think of that I will obviously I'll seek out anytime because they're just ruthless. But where like if you're talking about like I don't know your Twitter 3 for the cat or your next AI thing or whatever, like people are like, oh yeah, sure, that sounds fine. Like there's few people who credibly know things about it anyway, and it's not likely to elicit that kind of feedback. Where at a technical, science event, they're just like, yeah, outsider, like this is stupid, and they don't hesitate to do it. I'm part of the culture, I think, a little bit in sciences that they are more forward with that kind of thing, and yeah, I absolutely used it to my advantage. I had there was one person in particular who, after doing this more than once, eventually politely asked me to not talk to him again. Stop bothering him. But as I kept coming back, I'm like, all right, well, I think I solved this one. But yeah, it was a lot of that. And I mean, I just eat, sleep, and breathe medical journals, and it took, I don't know, probably 6 months or so before I could read those fluently. I used to have to read them, Google open next to look up all the words I didn't know. But so they have their own lexicon, and I think in some respects the lexicon I like to use, this parlance from computer science, was kind of offensive to the field back then, but has largely been embraced now. It doesn't seem so out of place anymore. I mean, think of like David Sinclair just wrote this book on information theory of aging or whatever, and it's like this is the party line from day one on Ocean. It sounded foreign now. Now you've got like Harvard scientists who are publishing on it whole books, and so I think the world in some respects evolved around us. Like back then, like I said, there weren't any gene or cell therapies approved, and when Provenge got approved, the dendritic cell therapy, ex vivo cancer vaccine was a big step because it was a manipulated cell therapy, and the rise of CAR T was huge, and once that started, like suddenly people started thinking, oh gosh, you could take cells out, modify them, put them back, and that can do some really cool stuff. And so we didn't actually change so much to the environment because the environment changed around us. And I think that's the actually probably one of the weirder things about the journey at large was that at some point along the lines, I crossed like this invisible line where I went from being a complete outsider to just someone who was there. Like, and it was a weird thing. Like, and to the point, like I became like, I guess, a rogue PI of sorts. Like I had my own lab, my own team did cool stuff. And you know, say, oh, there's like, Dr. Scholz doing something weird again. And I would have to like correct like, half the talks I've ever given. I'm like, I don't have a PhD. Like, I'm not a doctor of any sort. Like, then to edit my chirons and whatnot. But there was a weird line, yeah, where it seemed to happen overnight, where it had been for a long time, it's just completely an outsider, and then suddenly, yeah, not anymore. Maybe still viewed as crazy, but crazy and a part of the system.
Yeah, I think George Church talking about that coding parlance. Even going back like since the beginning of CRISPR, he's referred to it more like code, and I mean, it kind of acts that way. I mean, I do programming, so like it's like it is. I don't know how to do the in lab stuff, but in terms of like what I'm reading, it's like, oh, this seems very like one-to-one. And I like the neat thing is that people are more and more understanding code, like the world around them. But besides lower generations, I've been recently reading that people that are below millennial age, they don't know how to figure things out because and this might be one of those mean things, like all those young people, but apparently because everything's so well designed nowadays, they don't know how to troubleshoot as much. It's like when we were kids and we couldn't figure something out, you couldn't like Google how to solve it, so you had to like break and hammer and figure it out. Like the internet, for instance, didn't exist. I assume you're about my age. So like the internet did not exist when I was a kid, and so so apparently like younger people, because everything's like Apple beautiful, you just click a button and everything works. Apparently like troubleshooting is weirder for them or something. But I don't. That feels like one of those like older people being mean to the younger people. I'm sure they're good at problem solving too. But the which is just to say, like coding, I think as an analogy, yeah, is definitely more mainstream, and people like George Church and stuff as well. Where I think we're using it the whole time, which is kind of nice too.
Yeah.
So you have, like,
there's definitely like a subset of biologists that thought of this. I mean, one of those weird dissertations I'd read that I was super interested in early on was so I wanted to understand what was the key space of the humoral immune system. Like, what is that? Like, if you think of like DNA as base 4 assembly, a certain number of bits that encode the sticky ends of your B or T cell receptors, but they are not infinite. It's a bounded space, and so I was curious, like, okay, what if you're going to use a password cracker on this, for example? What is how many computations do you have to do to go through that whole space? And that was something like I found it on like a derelict server, and I'm pretty sure no one had gone through it in years, but it was written a long time ago. And so there was definitely like a thread through here that goes back to the beginning of the field, I'd say. But it just wasn't mainstream, and the average like molecular and cell biology crew did not like it. It was it's always been there, but it was definitely a minority, and one that was tolerated less when you weren't George Church, probably, but a renowned scientist that teaches at university.
What were any of the people that were original detractors? Were any of them eventually hired onto the team? Were any of the people like really? I guess the gentleman who said like stop talking to me. It'd be pretty nice to hear that like he like was on the board now or something. But
what's funny is he did find me like a few years later, and he came up to me. He's like, "Hey, I just wanted to say like I'm impressed by what you accomplished here."
Oh, that's nice.
I was like, "Oh, thank you so much for your help. Like you were super helpful early on. I appreciated it." But I think there were definitely, I mean, in terms of like investors and advisors and things like that, people who are like really raked us over the coals, who became really big advocates going forward. But I don't know if there's anyone who we hired outright who thought is entirely insane. But just because we weren't a real big group. But yeah, there's definitely been people who, I mean, in fact, I think some of the biggest advocates, we had over the years have been people who initially were very skeptical of it, and that once they kind of became convinced of it, they suddenly were like, "Oh my gosh, this is like I have an epiphany of sorts here. This is brilliant. We should totally do this."
Mm-hmm. But actually, speaking of investors, I wrote this down for that you were able to get Tim Draper to invest in Immusoft. And so I was just wondering, is there any story behind going, like, I think we're kind of talking about it now, like going from like complete outsider to getting someone who's like one of the key investors for like an entire space?
Yeah, well, so the way that initially started to go down was we became one of the first breakout labs companies, and so
Mm.
I mean, getting it funded was not easy, right? Like, say you want to build something that has never existed with no background in the space is not an easy sell. Like, it doesn't check a lot of boxes. And when Breakout Labs got started, they were basically, so this is part of the Teal Foundation, so Peter Thiel's group, and they were basically trying to fund things that were too early for venture capital and too outside the box for the NIH. And they issued a press release when they launched, and it's like, this is what we're looking for. And our actually COO today had found this, and he sent it to me. I was like, man, if these guys aren't going to fund us, no one will. Like, this looks like it was written for us. And so we applied to it, and we became part of the first cohort they had. And then it's how I got initially, introduced to Peter Thiel, and then Founders Fund led our Series A. And I'd met Draper at one of those events, actually. I think connected to that whole ecosystem. And so he came in the Series A also with Founders Fund.
When you're at an event like that, I think many people listening in, or, maybe nerdy scientists, maybe someone on the sidelines trying to transition in like you did, and they're seeing someone like that, and maybe they would come up and they wouldn't know what to say. What were like? Do you go up there and talk to someone like that with a strategy in mind, or you just say, "Hey, Matt, how you doing?" and then like start a conversation? Like I imagine that person's kind of being like locust a little bit too.
Yeah. I mean, with those guys, you really need to get, I think, an introduction to them. Like I've been involved in Breakout Labs for quite a while before I ever met, Peter Thiel, and they're like with Tim. I think that I can't remember the exact background for that, but I think a lot of them, like, mobbing them just at an event is tough,
Yeah.
and if you do, you have to have it down to like a 10-second elevator pitch basically, and just try to get a card or get a meeting, and maybe you get thrown to the gatekeepers or whatever, or you can say, hey, we met at this event, and I remember my app store for the human body, or my treating aging with information, or whatever little soundbite you can throw to stick in their head. But you have to lodge something really quick. And actually there's a I guess probably an okay story to share. Tim Draper, you'll laugh, I'm sure, at this, but so he invited me to an event in Vienna a few years ago in Austria, and he's like, "Oh, we'll meet here, and we can chat." Because I was trying to get him to put more money in, and so I go all the way out to this event that he gets me in. So a long trip for a small company, early CEO. And he was mobbed like this entire time, and I was having a real hard time actually pinning him down. And so he is going to head out pretty soon, but he had this big thing on stage, like it is a huge hall, like hundreds of people, and right before he got on stage, the newly elected is the prime minister of Austria. So it's like a high profile event, and then he gets on and does this whole thing. And I decided the best way to corner him was to sit in the front row of this audience. And then when he got off stage, I walked onto stage from the audience and slid behind the backdrop and captured him. And I'm like, man, if Secret Service is here, this is going to be the shortest little talk of my life. About to be tackled. But yeah, so sometimes you have to be a little creative to pin people down. But walking onto the stage from the audience is probably one of the more insane stunts that I've pulled over the years to meet someone, but and someone who I was supposed to be meeting with anyway.
Mm-hmm. Yeah, if I see you at a conference, I'll try to do that too. So, we've already met.
I
just to see your
face. Service, so it's probably no one's there to protect me.
But when you first were talking to him, and this is really a question about there's this quote saying that you're good at, like, if you go around telling people I'm good at X, it means you're probably okay at it. But if you go around and people are like, oh wow, you're good at this, it usually means you're great at it. So when you first were meeting this individual, or was he aware of you, or did you have to speak up for yourself? Like, was the fact that you were in the community and you took your time, like you said, like you were in the fellowship for a while before you met Peter Thiel or what have you, did your work lead the way for these successful relationships to be built, or? And that's fine if they didn't. I was just kind of curious.
Yeah, I mean, given that I was always such an outsider and didn't have like the credentials or anything to like be really, brandishing. I usually was more talking about like what I wanted to do and my vision for what I wanted to do, as opposed to like myself or why I was like the person to do it. In some respects, that there's a benefit to doing really crazy things is they haven't heard this pitch 100 times. They probably never heard this pitch ever. And so I don't have to be like, oh, I have like I'm the best person to build Twitter 3, like, or I'm the best person to make this new model. Like, I was the only one pitching that kind of thing. And so I was mostly focused on trying to convince them I wasn't insane. But I think that with a fair number of the kind of high profile investors I've gotten, like it probably had more to do with what other people said to them in terms of getting that meeting initially. Like these guys, could be meeting every minute of their life for the next 3 lifetimes. There's so many people who want to get FaceTime with these people that and if you're just like pestering them or telling them how cool you are, I don't know. I'd have a hard time doing that. Like the reason, like, I mean, I fear they want to talk to me anyway is because there's something cool that I'm doing that. Like, I mean, my goal is to make all these people a ton of money and to build something that no one's ever built. And so I don't think anyone, came there to hear about me. So I don't usually talk about it that much. Every now and then they ask, like, how the hell did you end up doing this? Like, that's usually the segue if it does come up.
And then just a quick question, because I think Oisin recently completed their seed round, if I was reading this right, or if my memory is serving me. I did not write this down. I should have for this question. Are seed rounds for biotechnology relatively boring? I've read the like in the sense of like, do you just use like a convertible safe, or is there other like ways of setting them up that you found to be more successful?
Yeah, I mean, this also is probably a little unique to me in that like there's a big, I'd say, bifurcation in bio between companies that come out of like your arch or your flagship or these big like formation entities, that huge like kind of Series A's and stuff out of the gate, and ones that kind of come out of spun out of academia with seed funding, and then probably more rare, or even ones like mine. And so we got a whole bunch of like angels and small VCs early on as we did this, and so we did a lot of things like safes and convertible notes early on. So we're actually doing Series A for Ocean now. We got a big pharma strategic investor to lead it, and which is also a little unusual. Those aren't the typical leads they often are following. But in our case, I thought it was a really good fit because they understand the science really well, and so they're yeah, I don't know if I'd use the word boring, but they don't do anything super exotic. They're more or less same kinds of things. And I know things like safes and notes are controversial in some circles, but one of the reasons I like them is the last person I want to screw over is the first investors. And if you do things like set evaluation that has no real basis in reality, with something that is like crazy high risk, high return in a market that could be very different in a couple of years, it exposes them to a ton of risk, and that they could just be obliterated due to like a change in shift in the market, or due to like something taking longer than it should. And so I think they have more protection in these things that allow them to just convert under more favorable terms in one way or another. But, so we've used those a bunch.
Yeah, I consider them more boring because I know people who will do like a seed round, and they will not do saves. They hate them or something. And it's like, I don't know, it seems pretty straightforward. They seem like the thing that you should do so you can focus on other things. And like you said, I think it makes it easier for investors to kind of invest in you, I think.
Yeah, I mean, they have flaws. I think they're not perfect. I mean, I could envision like a perfect document that was like a hybrid between a convertible note and a safe. I think would be like ideal. And I don't want to get into the legal business of just creating random documents. And in general, people tend to like use a YC safe or something. They're like, okay, I know what this is. You use a template that's as like from a major law firm and don't screw with it too much, as just the kind of way it is. But I mean, I understand like the strengths and weaknesses of them, but I think on balance they're a good thing to do.
So the back to Oisin and more science related topics. So this is pulling from MIT paper. In it says, and I can go longer if you need it, but the main core is in a new study, senior author Darren Baker of the Mayo Clinic in Minnesota found that if researchers removed senescent cells or prevented them from building up, they could prevent brain degeneration in mice prone to it. And there's more to it that I can give to you on this for context if you'd like. But I'm just curious what you think about that connection between senescent cell therapies and affecting brain health and aging.
Yeah, it's something we've seen like in our own studies actually. So we saw a cognitive improvement clearing senescent cells broadly in the body in old mice. And so they weren't prone to the disease, and it wasn't spreading in the brain specifically. But what's interesting about our approach, I'll contrast that, because I don't know if I've read that paper, but if they're clearing it in the brain, it's a little different. So like our typical vector doesn't really cross the blood-brain barrier. So it will transfect cells in the brain, but it's usually in the vasculature. And so even though we weren't like directly removing cells inside the brain tissue, we were seeing benefits in this. And I suspect that this has to do with just integrity of the blood-brain barrier and inflammation in the CNS. That you're clearing senescent cells across the whole body, and you're reducing this inflammatory state, and the body's able to function or regenerate better. The role of senescence inside tissues in the CNS is kind of it's a little different beast, and I suspect that you have more of a problem with on things like your microglia and stuff like that than you would with a neuron. But if you end up with inflammation and impaired garbage collection in the brain, it's probably going to do something bad. And so I think this, as a general thesis, is pretty defensible. But what I'm less clear about is which one is more important. Like if you improve the overall body environment, does that benefit the brain more than directly trying to target the brain?
Yeah, I think it was about a year or so ago, I had Oki O'Connor on the show, which corresponded with my first time seeing a neurologist. Yeah, where they scan your head. And it was interesting to see that like most things that affect your heart affect your brain. A lot, like, so if you can keep your heart healthy, generally keep your brain healthy too. So which is like probably an obvious statement to like people who like know more about this, but it was interesting to think how, if you exercise more, you start thinking a little quicker. There's so many studies.
Is huge.
Yeah, it's huge. Yeah, I just kind of took it for granted. Like, oh, I exercise, I feel better. And then and there's studies about like even old people when they have cognitive decline, if they do some aerobic exercise, it decreases the rate at that decline, and even in some cases, kind of bring it back. And so, I'm really curious to see some of these long-term studies going on. Because even though we're coming in and we're like, garbage collecting, taking all these senescent cells out, all these different therapies coming on, I'm curious, like, what does a peak human look like? And is it Brian Johnson? But like, what, if you do the right things your whole life, more or less, like, what would that look like?
There's certainly some interest now in things like hyperbaric oxygen therapy with vasodilators and stuff like that to just increase nutrients to tissues. And even though people who do kind of alternate both, they'll do hyperbaric oxygen therapy and then they'll do hypoxic training. And so they'll like stress the body on one and then saturate it the other. And this seems reasonable to me, to be honest. I don't know if like, how solid any particular data set is for it, or like, it's I'd say experimental, but it does make sense that like your vasculature beginning to fail impairs pretty much every tissue in your body, and aerobic exercise improves that health and oxygen saturation, all these kinds of things in tissues. And so this kind of just makes sense, right? If you end up with restricted blood flow to things, you get like all sorts of dysfunctions, and so the counteracting that is a logical thing to do across the board. The idea of our general inflammatory cytokines and things like that, like how big is the role of one versus the other, is I think an interesting question that is going to be teased out a little bit more. But I'm sure they both play a role. I don't have much doubt about that. I just don't know to what extent one might be dominant.
is there a future in senescent cells and the brain at Oisin? Do you see like a future where you guys are going to work on that, or is that like
Um,
kind of just like an interesting thing?
well, what's weird, so our lead program now isn't even the Senolytic. It's like our lead program now, it makes you build muscle with that exercise. It's the follistatin
Mhm.
therapy. I know I should update the website, but um,
We get into the fall
and
stat, and my next question involves fall stat.
yeah, so and I mean, this is partly just due to like the pharma interest in the regulatory path and all that. But I think that the way we're likely going to move forward on the Senolytic, just in terms of just like future of the science, is building better and better constructs that can target subpopulations of senescent cells more effectively and more precisely. So the best data we have so far is the one where we hit P16 and P53 together. That is definitely synergistic. It doesn't seem to do anything bad, but I think that as the field moves along, we'll probably find other pathways to target on it, and we'll be able to
Mhm.
further adapt those. And I mean, one of the big challenges facing the senolytic space, Oisin included, is finding a regulatory path for it. So I think most people in the field, even those who are just observing it, generally buy the argument that removing the accumulated senescent cells as you age is a beneficial thing. That it's it can address a lot of problems, but that's not the same as applying this to a patient population that has a phase 3 endpoint you can show statistical
Mhm.
significance on and go through FDA. And so that's become like the bigger issue was regulatory, not technical so much. And I think that's the ability to zero in on specific subpopulations or to make something more potent, more precise, more reliable in one area increases your odds probably of getting something approved. But on the other hand, I think that if you aren't clearing senescent cells across the entire body, you're likely not going to see a benefit to it. And I think this is really at the heart of why the Unity trial crashed. And they showed this in their preclinical data too. It worked in young animals, not in old, and it was still effective on target in the old. Like it removed senescent cells in the joint, but it wasn't able to overpower the burden across the whole body. And whereas if you take them out across the whole body, even if you're trying to focus it onto a specific target, you probably need the body as well. And I think as we go forward, it's going to be how do you build these better constructs that pick off all of the ones that matter, but that are going to work in one indication.
You mentioned Unity, and that actually is a fan question. Their name is Lunchbox, and they show up a lot. So Lunchbox, thanks for the question. The general question is, are there any lessons to be learned from that failure of the knee? I have no idea how to say that second word. I'm mildly dyslexic. In age-related muscular degeneration. What did you learn from that failure?
Well, I'm much more familiar with the osteoarthritis study and the knee than I am their age-related macular degeneration one.
That was the word. The O word was that word. Sorry. That was the word.
But yeah, I think if you look closely at the big paper that they were touting, it was in Nature, I think is where it was the Nature Science, one of these big journals. Before they went to clinic, the abstract conclusion said it worked great, but you start looking at the data and you've got a panel there where it shows that if you do it in an old animal, it doesn't actually improve the score. It's no significant change on it. And this shocked me, frankly, that went forward into clinic when, I mean, no one's that lucky. Who has preclinical data that comes out worse than the clinical trial? Usually we cure cancer in mice all the time. It's much harder in human. So why take it forward? And you don't have 3-year-olds with osteoarthritis usually. Not mediated by senescent cells probably, but and so I think that really was at the heart of it. Now I don't have any inside information on them or anything like that, but I know that the compound they're using is too toxic to be used systemically. I mean, it was effectively a failed chemotherapy compound, so they were kind of restricted to local administration of it. And in the eye, I think you could make a case that the eye is somewhat immune privileged in a restricted environment, and maybe clearing cells just in the eye would have a benefit, even in an old animal with a largely a SaaS-ridden environment. But and I don't know. Like I said, I haven't dug into that too deep, but certainly in the knee, even preclinically, it didn't work. So in an old animal, and it did clear those cells. I mean, that's what I guess it was on target effective. I don't think anyone had doubted that, and you could show certainly in an old explant it worked, but I think it's a broad environment that got it. And that's my general assessment of it.
This we're gonna get back to science in a second, but in an article by Fierce Biotech in 2013, they're quoting you on something, so I just wanted I'll set it up. The then CEO Matt Scholz spoke in skipping primate testing and going directly into humans in the next couple of years, hopefully. And this was in reference to immuno. So I'm a big fan of from the outside, as many animals being tested as possible, because of the terrible things that happen when you don't do it. What do you think about organ on chip, skipping primates to go straight into humans and stuff like that? Like, what do you think about that for clinical trials?
Well, it's funny. Like, I've never been a fan of animal testing. I mean, obviously, it's something I've done a lot of, and I mean, I've made such a point of visiting these facilities, seeing what I don't want to become detached from the consequences of my actions. But I think it's necessary at this point to do them. There's no way around it. I mean, it gets kind of preposterous at some point. You're like, you kill a human because you didn't want to test this on an animal. But the reason actually I didn't go to primates, I mean, I'm happy to not do primates if I can avoid it. But the reason that the FDA didn't make us do it, or we convinced them not to, was that the system we built didn't work in primates, period. And so that was an ex vivo cell therapy. And we couldn't even at the time make it work in a mouse. Like, we had to take an immune deficient mouse and then get them to support the human cell. So the system was entirely human from day one. And I tried quite a bit actually to make a primate version of it. And it just, I mean, there's weird things when you start doing ex vivo cell therapy, like, you can buy a cytokine from 2 different vendors. One will work, one won't. And you switch animals and they're like, oh, that's IL-2. And maybe it's something that was similar to IL-2. It got the same name, but you don't know if it's really doing the same exact thing in the body. And so we basically said, look, there's zero scientific merit for doing this. We can't modify the primate one, and the human ones will be slaughtered. They don't have we can't use an immune deficient monkey the same way we can a mouse. And so that was the rationale for it. And at Ocean we did primate studies actually very early, and the reason for that was that if you look at the history of lipid-based delivery systems, a lot of them looked really good in mice and become wildly toxic in primates and humans. And I mean, think of the history of these things, from what was basically something like a lipofectamine type compound to the modern LMP. Even the modern LMPs, when were the days when Moderna had IPO'd and stuff, and we're going to treat everything with LMPs. It'll be awesome. We're going to have cancer, all these things, and basically are relegated to the liver and vaccines, because if they administer any more of it, they're going to barbecue that liver, and they just become wildly toxic when they accumulate. And so we were aware of this problem, from the very beginning, and so we wanted to make sure that ours didn't have it. And so we were incentivized to show that we could administer very high doses to the primates without toxicity. But yeah, I think that at some point we'll be able to largely get around this. I'm a huge fan of the idea of, say, a person on a chip kind of testing. That there's certain things where it's hard to imagine it actually working for, how do you age your system on a chip, for example, or does it basically reproduce all these epigenetic changes that might be really important for a therapy like mine? But I think, to the extent that we can minimize animal testing is great. And in some of these tools, they're getting better. And I wouldn't be surprised if we saw a certain classes of molecules be kind of tested on them, maybe not exclusively in the short term, but I mean, the FDA has paved the way to do this, but fairly recently a policy change on it. I know, was it the Dave Goebel and those guys were working hard on this, and so I think it'd be very advantageous if we can get this to work. The more we can keep away from furry little creatures in cages, the better. But it is hard to get away from them.
I'll have to ask Dane that question. He's coming on the show in a couple of weeks. But as well. I'll ask, see what he thinks
Oh
as
yeah,
well.
yeah.
Yeah.
He'll love talking about that. He's working hard on it.
Yes. So, Barrel Master, which I think he's the person who is in the field or a PhD person, because he has very good. I don't know what their gender is. Ask great questions. So there's gonna be like 2 or 3 questions just from this person because they were so good.
All right.
When do you think 2 input and gate promoters will be good enough to target specific organs with your PLV?
Oh, I think actually pretty soon. It depends on the. So I guess I'd say from a technical point of view, it's a cell type, not an organ, that is the challenge. Like, because if you think of like in an organ, you could have like say a fibroblast versus hepatocyte or an adipocyte, all living in the same organ in the vasculature. And so if you're to be technical, like you're after a subpopulation of them, and I think some of them are a lot easier than others. That's just, I guess, an artifact of biology. I'll give you an example. Like we have a program to kill fat cells selectively, and it seems to work pretty well. A fun bit of trivia: there's a mouse model transgenic called the fat attack model, which is actually what the ink attack model is based on. That Baker Mayo Clinic one. So you can wipe out adipocytes, and it doesn't seem to cause any trouble. But I got interested in wanting to take out visceral adipocytes selectively. So not hit subcutaneous ones like your cheeks and your ass and all that kind of stuff. And that's proven very difficult. There's not like a single clear promoter to go after. And so then you do really need this more logic gate approach. And I said in some tissues, it's pretty straightforward. You could do it today without much trouble. And in others, I think the logic gets more complex. And it has to do with really just how the particular cell type is, what genes it's running. And there's some issues with we have limitations on how you study this with large transcriptomic databases or single cell RNA-seq type applications where is every cell there really the same? I mean, if you think of like your lymphocyte populations, we keep finding new populations we never knew existed at all. And so as you start looking at your organ, you think, okay, well, if I'm just trying to make the liver produce an enzyme, or say I'm making false enzymes, that's not a big deal. I can hit the liver, I can restrict it to the liver. That's easy. And if I'm off target a little bit, probably not going to cause much trouble. If you are carrying a suicide gene, you're saying, I want to kill just this one cell, and you mess it up, really bad things can happen. So I think there's not like a super easy answer to it. Part is we can do some of this today. It's not a challenge for certain tissues, and we can skew the biodistribution of the PLD quite a bit too to kind of aid that to some extent. But some of them I can't do right now that I want to. So, and I don't know for sure when I'll have a working version of that, but I think soon, like the kind of rise of like machine learning and control logic of the body in particular, is moving very quickly. And I think that some of these tools are going to allow us to build things that would have been really hard to build in the past. And I want to take advantage of that.
Mm-hmm. When it comes to your therapy, will it be? I imagine this can be some form of injection. Will it be like something that you would take like once a year for a period of time until things are better, and then you take it again when things go bad? Like, how do you see it actually when how it'll play in people when it's full through the clinical trials?
Well, the muscle building one is probably going to be done like once every 5 to 10 years, I'm guessing. And the senolytic one's different in that I think it will depend on your age. And so if you're really old and you're kind of frail and you have a huge senescence burden, maybe you had some cancer therapies in the past, you might be better served by taking a few lower doses and then going to a high dose of it. Other than before just slamming your body with really high dose of it. And but once you clear them, I mean, you think, how long does it take to get a wrinkle? It's an interesting question. Like your body accumulates senescent cells from birth, but it doesn't. They don't start causing dysfunction for a while. So and they begin to accumulate kind of exponentially as you get older. And the question, I guess, comes to if you knock them all down, like say you could wipe them out to zero as a 70-year-old. do they rebound at the same rate or not? How quickly do they come back? And what are the other kind of variables going to do to this? And so I would think like the D and Q therapies right now seem to be kind of moving to a stage where you do them like maybe once a quarter or once a year for your cycle of 3 days a week or whatever. I think it's the standard one now. But that's not a super good senolytic, admittedly. So the question is, like, you've got a really good one. I'm guessing you're going to need repeat dosing of it in the senolytic, no matter what. It's just but I think the exact schedule will probably vary by age and what you're trying to treat. And so we're looking at it for blocking the transition of acute kidney injury to chronic kidney disease. And in that scenario, it was probably going to be done as like, one dose every couple of weeks for, a couple of months, then you'd be done and you wouldn't take any more, because it was just trying to break a feedback loop that was causing trouble. But if you're looking at it for aging, I think you're pretty much stuck having to do it indefinitely, because you'll keep getting yourself.
What would for the muscle line, what would happen theoretically if you were to put it in just like a healthy population? Would it like juice them up at all, or would it? Work for people? Okay.
No, it should. Like our first studies in mice, we were just using normal, healthy mice, and it was one administration of it in 6 months, they were 50% larger and twice as strong. And so it's it absolutely should juice up a healthy person. In fact, one of the outstanding questions is, how frail can you be and have it still be working all right? Like, does it have is there a cutoff at some point where it wouldn't work? But it absolutely works really well in young healthy animals. And in fact, if you do it as a transgenic, the phenotype is 4 times the muscle mass. Massive. That's only been done in mice, to my knowledge.
Are there any downsides to the Arnold Schwarzenegger mice? Like, is there, I know with steroids, like you can have like heart effects and stuff like that.
Yeah, so in like follistatin therapy type context, there doesn't really seem to be. Like, the metabolic health is improved. We didn't run full lifespan studies on them. Some other people make claims it improves the lifespan of it, but I don't think they've been studied extensively. If you took like the transgenics that are 4 times the muscle mass, I think that probably causes enough stress on the heart to be problematic. Because if you were to scale it at the human size, 4 times the size of a normal human, you're still going to be weighing like 400 or 500 pounds. Like, and even though the therapy itself isn't acting on cardiomyocytes, the stress on the cardiovascular system may have an issue at that point. But and I don't think anyone ran those transgenic mice out for lifespan studies or anything like that. It was more just to explore the pathway. But yeah, I think that in the therapeutic context for follistatin, there doesn't really seem to be any downside to it. It would, I suppose, theoretically make you less resistant to starvation. So you're acting on a pathway that's built to conserve energy effectively. The reason myostatin is there is to make you as weak as possible, and it's I think largely thought for metabolic reasons. A healthy human can go for like a month without food, or a healthy man at least. And I think there's some sex differences between resistance to caloric restriction and starvation. But yeah, so I think that's a theoretical downside to it, but it was one that didn't seem likely in the context that we're living today. I think one interesting aspect of follistatin compared to, say, like a steroid or testosterone is that, like, follistatin isn't different between men and women. It's the same pathway. It doesn't have any androgenic effects. And so, testosterone is this kind of master regulator of all sorts of stuff. And follistatin doesn't really do that. And so there are different splice site variants to it, interestingly enough, that do different things. But those are not what you use in this context.
I can see this being like Ozempic, I think it is, where it like helps you with weight loss. Because one of the best ways to lose weight is just to have more muscle, because then you're just ambiently burning more calories, and then
Oh yeah,
like that.
This is of great interest to the pharmaceutical industry for really this kind of reason, and that if you take Ozempic or semaglutide or any of those GLP-1 type agonists, and you lose a bunch of weight, you lose muscle along with the fat. And I think the average ratio is about 60-40 of fat to muscle loss. And if you stop taking the drug, then the rebound effect is really directly correlated to how much muscle mass you have. So in some cases, you don't really have much of a rebound effect. Others, they'll balloon right back up. And so the thought being that, yeah, if you could pair something like that with something that drove a skeletal muscle growth, you would have a far more comprehensive metabolic therapy, and you'd reduce the risk of kind of a rebound effect, because it's not just that you're burning more calories, although I think that probably plays a role. It's that muscles regulate blood sugar and all sorts of stuff. So I mean, the reason we're so interested in that for old age is that, physical strength becomes everything. Like your muscle mass, protects you against falls, allows you to move around, socialize, increases quality of life, regulates metabolism. Like it's really a huge issue. I mean, makes you more resilient against disease or injury. And so it's playing a much bigger role than just vanity or just how much you can lift or kind of the mechanical properties of it. And so I think that, I think as a sideline that things like semaglutide are probably going to become the first broadly adopted health span improving drugs in existence. That these things, like if you start looking at the data for like whether it be cardiovascular health, colon cancer, like everything across the board looks really beneficial. And I think it shouldn't be super surprising that, especially in the developed world, like people get old, they get fat, they get inflamed, and bad things happen. And if you get rid of that, like that's positive on a whole bunch of levels, not just because you might have diabetes or just because you are, obese. And so I think that these things are going to become adopted far more broadly. And I mean, you see this already, of course. Like you can go to like any med spa in the country and get yourself a prescription for semaglutide, like get a knockoff of it. They're like prolific, but to the chagrin of pharma companies. But I think a logical step after that is, okay, well, how do we, instead of just getting rid of something that was causing trouble, build up something that's helpful?
Is that potential something that investors have been really excited about? Because I imagine if people see Ozempic as like this big thing, they're like, hey, this can like boost muscle and do all these other, and I don't know what Ozempic's original thing was for, but I know like
diabetes.
Okay, so if you can do the stuff that we're talking about, it sounds like you'll be able to have your cake and eat it too, in a sense of like lose weight, be healthier, because like your weight affects everything. Like if you're marginally obese, like your cholesterol is worse, then heart attacks are more likely, strokes are more like just more muscle burning more, and then people are busy. It seems pretty something that like people with money would be. I love your glass.
Oh yeah,
it's a great glass.
People are pretty excited about it, I'd say. I mean, it's why it's become the lead program now. In the end, like pharma wants it, and that's I mean, it's important. Like if you want to build therapies and after longevity and health span, but like more than just making people live longer, you want to live better first. Like lifespan is a side effect of health span, and I think eventually, if we're talking about like, can we double our average lifespan again, like we did in the last 100 years, we're going to have to change the person, not just the environment. And so we're going to need tools that are, pretty invasive in that respect, like that they're actually altering core processes in your body. But, and I think that's great, but no one wants to live to be 200 as a 100-year-old. You want to live to be 200 as a 30-year-old. So making your body function better is kind of this logical path. And it's, in some respects, we've built this strategy around saying, okay, we want to make a drug, a therapy that can be approved by the FDA for something. I don't want to just like throw myself on the rocks of FDA should let me treat aging and longevity out of the gate. Let's just build things that have a known indication and that we can use for improving health span and lifespan. And then I think they will come around to this. But yeah, from just a business point of view and a strategy point of view, I think it makes a lot more sense to start throwing these in and collecting these data points. If you got, a licensing deal worth a few $1 billion and a product going into people, and you say, oh, I think we can use this for aging. That's a lot easier sell than saying I'm going to build something that no one's ever tested and use it in a human and say I want to use it for aging.
And then you can like split off some of that money and do more research and make out more IP. So
yeah,
is there what is so for the FDA from like the normal clinical route outside of just weight loss and being healthy? What is the target population that you guys are going for? Like, who are you going to be helping?
I said that right now on the muscle building one, we're looking at nursing home patient populations with just markers of sarcopenia and frailty. And I think we are still, I guess we're early enough in this process that it's a conversation we'll have with regulators about like how exactly to pin that down, but that's the gist of it, because they're in a controlled environment. Like you can watch them, you can measure them, and ideally can show lots of benefits over time. The other route we've looked at, and we haven't I don't know, we still might go after it, but it's like one that we hadn't put as much into is actual local administration of it. Because if you put it into a muscle, that muscle gets bigger. It's noticeable. And that of course is of interest for cosmetic applications, but also it has a huge issue in like injury. And I think that like if you're immobilized, you can still use this, for example. And I think that's something we looked at, but we prioritize the systemic one because frailty is more kind of on mission for the longevity focus, where in local is has some benefits, but it's easier to go from systemic to local than from local to systemic also. So.
If you does the therapy have any effect on healing? Like if I like you just said, like injury. So if I tore, like you tear muscles and just exercising, but if you do damage to it, is there like any effects on regeneration at all?
I think so. There were some early studies where they're using things like snake venom that was myotoxic and stuff like this, and showing that you could heal faster with follistatin therapies. And I think from normal injury from exercise and use, you see recovery faster because it's stimulating muscle growth. But in the case of if you say broke a bone, it would prevent the atrophy of the muscle, or should mitigate it substantially. But I don't know if it would have any effect on the healing of the bone or healing of skin, for example. I don't think it's like broadly speaking pro-regenerative. I think that it enhances regeneration in muscle specifically.
Mm-hmm. Yeah. So I'm a big fan of SENS. Love Michael J. Fox type foundations that basically create IP, which then gets licensed out and does great things. How do you think, as someone who's with your experience, what can be done to encourage and support these kind of like Bell Labs types organizations that are doing this type of work?
Yeah, so I mean, interestingly enough, my co-founder had started a foundation, the Long Range Institute, to basically accelerate longevity therapies. Also, as we built this, and largely on this translational aspect to try to make them available faster to people. And I think, I mean, where you see probably the most of these kinds of foundations that prove to be effective are in the rare disease spaces. There's a somewhat morbid quip that the best thing that can happen to a rare disease patient is for a movie star's kid to get it or something like that. Basically someone in Hollywood who will publicize this, and then you get like people mobilizing around it. And I think they can be very effective at taking things that would otherwise die in academia and advancing them to the point that a pharma might find them interesting. And this has been, historically a problem. It's why, the Orphan Drug Act and all that stuff was created. That, if you're going to spend, it costs basically the same amount in R&D to make a drug for 1 million people as it does for 10. And you can't recoup the same costs from a small population. So that all these incentives were built to try to encourage development in that space. And I think with a good deal of success, really. But with the foundations, like, rarely have, I think, the ability to take a drug forward the way a pharma or a biotech startup would. And they typically don't have the academic resources themselves to make like really like new deep science stuff. So I think they typically are most effective as kind of facilitators of sorts, where they're like sitting there saying, okay, we're gonna find these people who are working on it. We're gonna give them more money so they keep working on it. And we're gonna take this IP and we're gonna figure out how to like shop it to a pharma or how to get it over this wire. And if they can't get a pharma to do it, we're gonna talk to like an academic hospital who will do it as an investigator-sponsored trial to like get a human proof of concept on it and publish this. And so I think that's how, at least just from my as third party observation, I've seen them tend to be the most effective. I think you could definitely do. There's other approaches to it. Like I said that Gary Hudson, the co-founder, his idea was to use it more as a translational tool, or like I guess later in this process, where you take something that seemed to like work and apply it to make it accessible, or accessibility tool, as opposed to a straight development one. But yeah, I guess for the purposes of your question, like, what do you want to actually support? I guess it'd be like if the question is how do you support these groups or make them better? What do you want the group to do? Because I think the answer would differ a bit. Like I'm describing how I've seen them most successful, not necessarily how to directly answering your question.
Yeah, I guess the answer would be to affect positive change to diseases. So, not just like generate IP, but IP that can help people and improve their lives, like diseases and stuff like that. I think Michael J. Fox, like I was talking to someone, and they said that his organization is basically the reason that they might have like actual Parkinson's, like massive, like the world with or without that foundation is the difference between a Parkinson's being treatable, like something we can do about it, like pretty quickly soon from now, and maybe this century.
Well, and think about that. Like, I was back to my cynical little quote that because you had a famous person who got it, who had the ability to publicize this and serve as could attract this level of interest from science, like they were able to serve as like a focal point or a group that could gather others together. And that's effectively what the foundations do. Like, and I mean, Michael J. Fox is a pretty big foundation at this point, relative to other rare disease ones.
Like 500 million,
yeah, I mean that's a good size, and so they're capable of doing things that others couldn't. But I think if you look at like what is it that made the difference in that, it was at some point some scientist who was messing around with these kinds of symptoms, heard that, oh, there's someone interested in this. I bet I can get like my lab some money. I bet I can make another paper out of it. And those are always the earlier actions that take place. Like I think that a lot of the world's problems have been solved, and the answers are just trapped between the ears of a scientist in a lab. And there's a huge problem, I think, with academia, and that like the currency of academia is really publications. And so you get a grant, you write a paper, and typically by the time you apply for a grant, it takes a while to get it. By the time you get it, you've probably already done the work. You use the money on something else, you publish the paper, then you go on to the next thing. And that's the value. Get the paper, like high profile, high impact journal, and do something else. And that it's not an organization incentivized to build therapies. And that's why, like, I mean, when ImmuSoft's a good example of this, actually. So ImmuSoft's core technology came out of Caltech, from a Nobel laureate's lab, David Baltimore's lab. And so it's as high profile as you can get. The guy was president of Caltech. And I had met him randomly at an event, back to our earlier conversation, and said hi. But I called him up and was like, hey, I want to program B cells, and you built this cool lymphopoiesis system. I think I can use it to build my app store. And that sounds fun. Like, I'll connect you to tech transfer. And that was like this start. But thinking back, how someone with no background in bio at all could stroll into the Nobel laureate's lab and make off with that technology for very little money was just a testament to how underexploited that space is. That there's not enough people who take things out of the lab into the real world. And this is why I'm so passionate about that niche. Like, it's full circle on this. And foundations, I think, are capable of operating that way. Like, and the ones that are successful often do, where they're doing kind of what I was doing, but doing it as an organization, not as an individual, and focusing more on usually, like, when the foundations get started, there often isn't a clear path to what you want to do. Like in my case, I had a very clear idea of what I wanted to do from the beginning, and I was just looking for pieces. But yeah, there is so much latent potential in academia in the U.S. just begging to be exploited. And I think I've had largely good receptions from scientists for it, even ones who, like, looking back, I wonder why they talked to me at all. But I think they want to see their work do something good too. And they say, hey, I think I can use this to cure some horrible disease or to make someone's life better. And they're usually receptive to it. And so that's something I think that if you're building foundations for a particular cause, one of the first steps you take is you identify, who needs this thing and who's working on this thing, and you try to bring those people together, and then collect enough money to make something go forward that would have otherwise been forgotten.
Actually, if I could have a quick question, is there any help that you need, as we have everyone listening in? What help do you need and Oisin needs to achieve this objective? Like anything at all that people listening could help with?
Well, I guess my party line is I'm usually looking for money, brains, and molecules. Always trying to I guess connect smart people and ideas of resources. But I guess more specifically these days on the science side, I'm still very interested in manipulating the control logic of life, and so like these ways of designing new Boolean logic. I think that this marriage of computer science and medicine and synthetic biology is really where the field is going, and I'm really interested in bringing those things to bear in longevity, and I think they'll enable us to do more and more powerful things with the delivery technology in particular. And so anyone who's interested in that kind of stuff, I'm always excited about. People who want to do clinical trials or have really cool models and they want to play with them, that's another one that's really common. And obviously, we're raising a Series A, so anyone who feels like writing checks is always on the warpath for that. And in the rare event that I'm not raising money, I always know someone else is. So that's I suppose a common one. But yeah, I think I really I think all the greatest innovations happen when disciplines mix and you have these ideas and two different perspectives come together. And so I really enjoy interacting with other people who are approaching these problems with different tools and different ideas, and think about how to combine them.
Thank you so much for sitting down today to be on the show. I appreciate just you coming on today.
Oh, well, thanks for having me. It's a blast, and happy to chat anytime. And I always love to get the word out and encourage the next generation of people to come on and join us in this battle.